To my class students, I am very much thankful to Department of Higher Education, MP, for providing me this opportunity to meet students all over MP. Myself, Dr. Alka Varma, Professor of Botany, Sarojini Naidu Government Girls PG Autonomous College, Shivaji Nagar, Bhopal. So, description of the topic. We are all students of Botany. Now, your class is M.Sc final. Paper is 2. The name of paper is Molecular Biology and Plant Breeding. Molecular biology, as you know, this is the branch, this is the science which deals with macromolecules and biomolecules like protein, carbohydrate, nucleic acids. And plant breeding, actually this is the purposeful manipulation of different plant species to produce a more good character. Now, this plant breeding is done by genetic methods also, like hybridization methods. But here, in this particular paper, these molecular biological techniques are used for improvement of plants. So, it is included with this paper. Now, prerequisite. What is prerequisite? Now, my expectation from you students are you should know nucleotide, polynucleotide, helix, antiparallel, occurrence of nucleic acid, and many other linkages. And the objective is what I will give you. This will be the overview of chemical structure of DNA, three-dimensional structure of DNA, and forms of DNA. Now, this is the three-dimensional structure of DNA. Students at this level, they know what is the backbone, what is this ladder-like structure. So this is a backbone formed of phosphate and sugar, and these are bases which join by a hydrogen bond. Now the synopsis, क्या पढ़ना चाहेंगे हम इसमें? The introduction is general about the nucleic acid, then history, which is very interesting, and every student of molecular biology or who is going to study this DNA should know very interesting history of DNA discovery. Then Watson and Crick. Every student who knows DNA is very much familiar with the Watson and Crick, this name. And actually Watson and Crick, they have given this model after much work was done on this. Then chemical structure of DNA, because this is a chemical and a macromolecule. Then dimensions of DNA, because it has its length, it has its breadth, and many other things in many other molecules it contains, and how they join each other, and what is the distance between them. This is called topology or dimension of the DNA helix. Now this DNA, which is basically consists of polynucleotide chain, it takes different forms because it is a coiled structure. This coiling may differ and give different forms of DNA depending upon the physical condition. Now there are circular DNA in prokaryotes and their coilings. And last is the point to remember. What are the important points you should remember? Now this nucleic acid. Nucleic, it comes from nucleic because it was first identified in the nucleus. Acid, because it contains phosphate of the phosphoric acid, which imparts a polarity, a negative charge, and so it is acid. This nucleic acid is the overall name for DNA and RNA. DNA, deoxyribose nucleic acid, RNA, ribose nucleic acid. Actually, nucleic acids are the class of biopolymers. Biopolymers, biologically synthesized. And polymer because these are macromolecule composed of many monomers which are called nucleotides, and nucleic acid, they carry hereditary information. Some cases RNA also. Now history is very interesting. In 1868, Friedrich Mischer, a Swiss biochemist, he discovered it and called it nuclein because it was associated with protein. He was studying the composition of lymphoid cell, which is WBC. Then comes an era when Mendelism was rediscovered. Then in 1903, chromosomal theory came into existence by Sutton and Bori. 1910, Albrecht Kossel identified nuclein and he gave the name DNA, nucleic acid. Moreover, he isolated 5 nucleotide bases which are building blocks of DNA. For this, he was awarded with Nobel Prize. 1928, a very interesting thing happened when the Frederick Griffith, his transformation experiments, which proved DNA a genetic material. And then came Morris Wilkins and Rosalind Franklin. These two names are very important because they first proposed helical structure for DNA by their X-ray crystallographic techniques. And James Watson, American biologist, Francis Crick, an English physicist. Watson and Crick in 1952 gave double helical, twisted and ladder-like model of DNA. Now these are the photographs of it. Students को ये चीजें पढ़ना चाहिए. Friedrich Mischer. Then comes the name of Albrecht Kossel, Rosalind Franklin, Morris Wilkins, and J. Watson and Crick. Watson and Crick proposed double helical structure of DNA. This structure is made up of 2 polynucleotide chains connected by a hydrogen bond. These chains run in opposite direction, that is, they are anti-parallel. Opposite direction, but it gives polarity, which we'll discuss in next slides. Each chain is composed of monomer. This is polynucleotide, so the monomer will be a nucleotide. This is the structure as proposed by Watson and Crick. This is the ladder-like structure having this backbone of sugar and phosphate, and this structure having nitrogenous bases. When this coils, this looks like this. Again it coils around an axis, gives a double helix to the structure. Now, if we see polynucleotide chain, which are formed of monomers, which are called, each monomer is called nucleotide. Nucleotide made up of nucleoside and phosphate, the component of the nucleoside and phosphate. Nucleoside is having a pentose sugar and nitrogenous base, and they form. These pentose sugars are ribose sugars. And ribose sugar in RNA, deoxyribose sugar in DNA. Now this is the structure of pentose sugar. So we start from here. This is the first carbon, second carbon, third carbon, fourth carbon, and this fifth carbon is out of this ring. This is ribose sugar because it is having an hydroxyl group at the second position, and this is deoxyribose sugar because it is not having any oxygen at this position. Deoxy, where oxygen is not present at that particular position. Now this is the structure, same structure, which is having 1, 2, 3, 4, 5 carbons. This is a very important structure at this first carbon of the sugar and third carbon and the fifth carbon. ये स्टूडेंट्स को ध्यान में रखना चाहिए. पहला कार्बन जो है वो बेस से जुड़ा होता है, नाइट्रोजनस बेस से. थर्ड एंड फिफ्थ कार्बन जो बैकबोन है फॉस्फोरिक एसिड की, उससे जुड़ा होता है. इसी से हम उसको पोलैरिटी कहते हैं कि एक साइड 5 प्राइम है, दूसरी साइड 3 प्राइम है. इस पूरी चेन में हम आगे देखेंगे कि कहीं ये 3 फ्री रहता है, कहीं ये 5 फ्री रहता है. तो ये जो 5 कार्बन जो फ्री होता है इसमें फॉस्फोरिक एसिड है, और ये जो 3 फ्री होता है इसमें OH होता है. तो हम कहते हैं 5 to 3 एंड दिस runs anti-parallel. Now, these are nitrogenous bases. Two types of nitrogenous bases, pyrimidine and purines. Pyrimidine is a single ring. These are single ring compounds. Pyrimidines, these are double ring compounds. Pyrimidines are thiamine, cytosine, uracil. Purines are adenine and guanine. This is the methylated compound. Also, this adenine is also a methylated compound. This 3 carbon of pyrimidine and this 9th carbon of purine, they join to first carbon of pentose sugar. Remember this thing. This 3 carbon of pyrimidine and this 9th carbon of purine, they join to first carbon of sugar by glycosidic bond. This bond is glycosidic bond. Now comes the structure which forms the backbone of DNA. Now this is H3PO4, phosphoric acid. As its structure shows, it contains 3 monovalent hydroxyl groups and a divalent oxygen atom. 2 of the hydroxyl groups, they are utilized in the formation of diester bond. Through this, these ester bonds or ester linkage, they link this phosphoric acid to sugar. And the carbon, as I said you, third carbon of the sugar or fifth carbon of the sugar, it contains phosphoric. Now this phosphate group, this is the structure of nucleotide. This phosphoric acid, this is the fifth carbon and this first carbon to the third carbon of pyrimidine. This is purine. Now, how these diester bonds are formed? This is pentose sugar in the DNA. The third carbon, hydroxyl group of the third carbon, it forms a ester linkage with the OH group of this phosphoric acid. The second OH group, it forms the OH group of this fifth carbon. These are 2 esters, so it is called diester bonds. Yeah, phosphodiester bonds. Here we see this fifth carbon is having, these are 1, 2, 3, here bases are there, and this first carbon of the sugar is involved in forming glycosidic linkage with the nitrogenous bases. Now we see this fifth carbon is having phosphoric acid. This third carbon is having OH group. This is what we call polarity from 5 to 3 direction. And this anti-parallel, which is parallel to this, it will form 3 to 5. Now, adenine always links with thiamine and guanine always with cytosine. There are 2 hydrogen bonds. These are 2 polypeptide chains which are anti-parallel. Look at this orientation of sugar. Okay, and there are 3 hydrogen bonds between guanine and cytosine. Here, thiamine is absent in ribose sugar RNA. You all know their uracil is present there. Now it is completely showing 5 to 3 direction in both. After looking at nucleoside, nucleotide, polynucleotide, certain generalization were made after analyzing DNA samples from different organs of the same body or from different bodies. It comes. Number of adenine molecule always equal number of thymine molecule. Number of cytosine molecule always equal number of guanine, because adenine pairs with thymine, cytosine pairs with guanine. But the number of adenine molecule did not necessarily equal that of guanine, and similar relationship was observed between cytosine and thymine. Double helix of DNA, 2 polynucleotide chains are coiled. Now, what is there? This double helix of DNA, क्या है? 2 polynucleotide chains are coiled about the same axis in such a way that they can separate from one another only by uncoiling, and that lateral separation is not possible. आप दोनों polynucleotide chains को खींचकर बाहर नहीं निकाल सकते, क्योंकि ये कैसा है? A double helical structure. Now, during coiling, the 2 strands, sometimes they come very close together, and sometimes they are very far from each other. During this time, this is called minor groove and major groove. Minor groove exposes less number of nucleotide, and major groove, because both the arms of the ladder, they are very far from each other, so bases are very much exposed. So a student who is studying this DNA should know. Now, complementary base pairing in 2 polynucleotide chains within an opposite. ये हम देख चुके हैं कि दोनों ही कैसे हैं. Yes, one word is there. You should know complementation. If one polynucleotide chain is having adenine, then the parallel will have thymine in front of this. X-ray crystallography and chemical analysis studies of DNA molecule established following features. Now, actually, this is the topography of DNA. Bases are set at right angle to the long axis. Everyone knows what is long axis. Up here comes a new thing. The distance between the 2 base pairs is 3.4 angstrom. And there are 10 base pairs in one turn. And this helical structure has a dimension of 20 angstrom. So it has a pitch of 34 angstrom. Now, what are all this? A double helix model of DNA is proposed by Watson and Crick. The topology or the physical dimensions are like this. It has a 20 angstrom diameter. This is called pitch. When it completes one turn, this whole distance is called pitch. This pitch, according to Watson and Crick, 34 angstrom in length, and it contains 10 base pairs. इससे इस दूरी को टर्न या पिच कहते हैं, और इसके बीच में 10 बेसिस हैं. प्रत्येक बेस के बीच की दूरी 3.4 एंगस्ट्रॉम है. अभी जो रीसेंट स्टडीज है उसमें यह है कि 10.4 तक बेसिस होते हैं इसमें. डीएनए, बीडीएनए, सीडीएनए, इवन ईडीएनए आते हैं. The helical structure of DNA which was described on Watson and Crick, was right-handed and is called B form of DNA. Other forms A, C, and very rare D and E forms. These all forms, A, C, D, E forms, all are right-handed. They show right-handed coiling. Z-DNA has left-handed coiling. And these alternative forms of DNA or the helicals, actually these are double helical structure. They show different forms. And what is the basis? किस आधार पर या क्या चीजें हैं जो इन्हें अलग करती हैं? तो पहला होता है नंबर ऑफ रेसिड्यूज. रेसिड्यूज मतलब न्यूक्लियोटाइड्स पर टर्न. जैसे आपने बी डीएनए में देखा, पर टर्न रेसिड्यूज है 10, इसको नाम दिया N. A second is the spacing of residues along the helical axis. What is the distance between these two residues? अगर डिस्टेंस ज्यादा है तो नेचुरली ये जो डीएनए है ये शो. नाउ इट इज प्रेजेंट इन डिफरेंट फॉर्म्स. जैसे हमने कहा डीएनए इज प्रेजेंट इन डिफरेंट फॉर्म. और वी विल speak about the coiling pattern of DNA. Actually, the model which was proposed by Watson and Crick, this DNA is called B-DNA, or it is right-handed coiling. It shows right-handed coiling. This it is clockwise coiled around the axis. Left-handed, it is anti-clockwise present around the axis. Now these different forms are जो structure, ये है different forms. अब इसको समझने की जरूरत है. ये these are the conditions when A, B, C, Z forms are seen. Now base pair per turn is 11 in case of A. In case of B it is 10. cDNA 9.33 and this zDNA shows 12. इससे हम कैलकुलेट कर सकते हैं कि ये जो वर्टिकल राइज है, इसमें वर्टिकल राइज मतलब द डिस्टेंस बिटवीन टू बेस पेयर्स, कंसेक्यूटिव बेस पेयर्स 2.3 एंगस्ट्रॉम. इन दिस केस 3.4 एंगस्ट्रॉम. वी से 34 डिस्टेंस है एक पिच का या एक टर्न का. सो जो 10 पेयर्स हैं उनके बीच की दूरी 3.4. ऐसे ही हम इसको देखें तो 9.33. सो इट इज 3.3 एंगस्ट्रॉम. देन 12, दिस इज अ लार्ज नंबर. एंड वर्टिकल राइज पर मतलब प्रत्येक जो है एक दूसरे से 5.7 एंगस्ट्रॉम दूर है. अब अगर हम इसके डायमीटर को देखते हैं तो यहां पर डायमीटर हमें दिखाई देता है जो A का है 25.5. दिस कम्स सम 20 एंगस्ट्रॉम हमने बताया, लेकिन 23.7 20 एंगस्ट्रॉम एंड दिस इज 23.7 एंड दिस इज 18.4. अब आप इसको एक तरीके से सोच सकते हैं कि जब डिस्टेंस ज्यादा है तो इसका डायमीटर कम हो रहा है. दूर दूर प्लेस्ड है ये, और एक्सिस से भी ये हमको इनकी दूरियां भी अगर हम देखते हैं तो इसका मतलब है कि यहाँ पर जो न्यूक्लियोटाइड्स हैं वो एक्सिस के काफी करीब हैं. अब ये जो डीएनए है डिफरेंट बिटवीन दो डीएनए बहुत इंपॉर्टेंट है जो zDNA फॉर्म्स, zDNA एंड bDNA. zDNA is left-handed helical sense, bDNA is right-handed helical sense, and phosphate backbone follows a zigzag path. ये हम अभी नेक्स्ट स्लाइड में देखेंगे. Here phosphate backbone is regular, they are in one direction. Sugar residues have alternating orientation and repeating units like this. This is zDNA. Phosphate backbone is like this. It is regular. Here it is zigzag. आप देखिए यहाँ पर जो दो शुगर्स हैं they are oriented in one direction in one polynucleotide chain. Here, these two sugar molecules, their orientation is different. Therefore, in this case, what happens? These two nucleotides, these two sugars and this nucleotide, they form one monomer. Here, one nucleotide forms one monomer. But in this case, two new. Therefore, there are 6 bases. These 6 bases are present in one turn. And because they are made up of every base, every monomer is made up of 2 bases, so there are 12 bases. And here, these are oriented regularly. And here they have different alternative orientation in this case. And this phosphate is showing zigzag structure. Here it is a regular structure. These are A form, B form and Z form. Here you see this is having more diameter. Bases are, because 11 bases are present there, so they are very near to each other. This is intermediate between both of these forms, and this is somewhat thicker Z form DNA, and it has less diameter. इसके डायमीटर से इसका डायमीटर कम होता है क्योंकि बेसिस एक दूसरे से काफी दूर ओरिएंटेड होते हैं. तो जब हम बात कर रहे हैं इस डीएनए की, जो कि स्ट्रेट डीएनए है, तो हम सर्कुलर डीएनए के भी उस पर भी एक सरसरी निगाह डालें. वो ये है कि ये सर्कुलर डीएनए प्रोकैरियोट्स में बढ़ रहा है. ये अपने आप में कॉइल स्ट्रक्चर है, बट इट गिव्स सुपर कॉइल स्ट्रक्चर ऑल्सो. When it is clockwise direction, we call it positive, and when anti-clockwise, this is called negative supercoiling. Now you should remember some points. A nucleotide consists of nitrogenous base, purines and pyrimidines, a pentose sugar and one or more phosphate group. Nucleoside is pentose sugar and nitrogenous base. Phosphodiester linkages are found between two nucleotides, between 5 OH group of one pentose and 3 OH group of the next. Complementary base pairs AT, GC, they are formed by hydrogen bonding within the helix. Glycosidic bond linkages between nitrogenous base and sugar group. DNA exists in several forms. Pitch and turn of DNA. What is pitch? What is DNA? One turn. Now there are questions. Write short notes on. I don't have to repeat this thing. This polynucleotide chain. You can write different dimensions of DNA model where we will give topography. Not only what's in Greek D model, but also the Z. Major group, minor group, supercoiling. Again, bDNA, zDNA, different forms of DNA, double helix, comparison of bDNA and zDNA. Many questions arise. For further principles of biochemistry by Nelson and Cox, very good photographs and colorful photographs and bright photographs. Then molecular cell biology, David Baltimore and Hobb. Molecular biology, David Fressfelder. Molecular biology by P.K. Gupta. ये बुक अवेलेबल है बहुत आसानी से. यू कैन परचेज ऑल्सो. रस्तोगी प्रकाशन. वेरी गुड डिस्क्रिप्शन एंड वेरी गुड डिस्क्रिप्शन और बहुत अच्छे से काफ़ी डिस्क्रिप्टिव है. सेल बायोलॉजी, जेनेटिक्स, मॉलिक्यूलर बायोलॉजी, evolution and ecology by P.S. Varma. These books are, and मैं तो एक बात सजेस्ट करूँगी कि अगर आपको बेसिक स्ट्रक्चर नहीं मालूम है तो आप सबसे पहले अपने अंडर ग्रेजुएशन लेवल की किताबों को पढ़िए, बेसिक स्ट्रक्चर जानिए, और मुझे लगता है कि मेरा ये लेक्चर आपको काफ़ी हेल्पफुल होगा यह देखने के लिए कि कैसे ये पॉलीन्यूक्लियोटाइड चेन्स एक दूसरे से जुड़ी होती है और कैसे इसके जो डिफरेंट मॉलिक्यूल्स इसको फॉर्म कर रहे हैं वो किन लिंकेजेस के द्वारा जुड़े हुए होते हैं। Now, thanks for watching the content. It will be helpful for you.